Today's news on Mel TV. I'm your host Leo King, and today we'll discuss how people can't keep a balanced diet simply because they can't distinguish the difference between food and nutrients. Today, to help us clear this issue, we have Dr. Petit. Good afternoon, Dr. Petit. Thank you for joining us. How can you help us clear this issue?
Well, in my zone of expertise, I can tell you that food is any substance, whether processed, semi-processed, or raw, which is intended for human consumption and includes drinks, chewing gum, and any substance which has been used in the manufacture or treatment of food, but does not include tobacco or cosmetics. Now, nutrients are any substance obtained from food and are used by the body to provide energy to regulate growth, maintenance, and the repair of the body's tissue. Now, proteins, lipids, carbohydrates, minerals, vitamins, and water are considered nutrients.
Interesting. So you're saying in your zone of expertise, can you describe the chemical composition of these things such as lipids, carbohydrates, and nutrients?
Yeah, you have to see. Lipids can be categorized as fats or oils, which are triglycerides formed from 3 long-chain fatty acid molecules and 1 glycerol molecule. The simplest carbohydrates are monosaccharides. They contain a carboxyl group and also 2 hydroxyl groups and have the empirical formula CH2O. Now, monosaccharides are the building blocks for disaccharides and polysaccharides. And you have to see, proteins are made up of 2 amino acids.
So amazing. Well, thank you, Dr. Petit, for your expertise, and we'll be right after commercials. Welcome back. Thank you for joining us on our second segment on today's news on Mel TV. I'm your host, Leo King, and in the previous segment, we discussed with Dr. Petit about the physical composition of lipids. In this segment, we'll essentially focus and discuss lipids in the forms of fats and oils. Thank you for joining us, Dr. Petit. It's always a pleasure to meet you.
Merci. Thank you, American, for having me here today. Well, to get the discussion ready, I have to inform you that when it comes to fats and oils, the naturally occurring fats contain a mixture of saturated, monounsaturated, and polyunsaturated fatty acids with different chain lengths. They are classified according to the predominant type of unsaturation present.
Wow. So, by any means, is there a predict the degrees of crystallization or melting point fats from their structures?
Well, you see, solids or fats are most likely to be crystallized, more saturated, and or have longer fatty acid hydrocarbon chains. More unsaturated, like olive and peanuts, have the and polyunsaturated fats like palm and coconuts are solid at room temperature. The melting points of fatty acids increase with increasing relative molecular mass and increasing degree of saturation. Fats and oils are chosen for cooking and the basis of their melting temperature. Now, cis fatty acids, those of which have the hydrogen atoms on the same side of the carbon-carbon double bond, have lower melting points than trans fatty acids, which are where the hydrogen atoms are on the opposite sides of the double bond.
I remember that from chemistry. Well, so which fats are more stable? Which ones do you think?
I think saturated fats are more stable than
You
unsaturated
know?
fats. I know this, sir. Don't doubt my intelligence, please. The carbon-carbon double bond, its unsaturated fats react with oxygen, hydrogen, lights, and enzymes, heat, or water.
Interesting. Doctor, can you describe the process of, I believe it's hydrogenation?
Repeat with me.
Hydrogenation. Thank you. Of unsaturated fats.
D'accord. Hydrogenation is the process of adding hydrogen to the carbon-carbon double bond of a fatty acid in the presence of heat at around 140 to, say, 225 degrees Celsius. Pressure, yes, we don't use American systems. Pressure and finally dividing metal catalysts like zinc or copper increase the amount of saturation, which can be partial or full.
Well, thank you, Dr. Petit. Anything you'd like to add about this say it again with me. Hydrogenation. Fats and oils?
Oh, of course. Yes, there are pros and cons for this process. Some advantages include decrease in the rate of oxidation, increased hardness, control of the feel and plasticity, and phase change from liquid to solid to make the melting points of an unsaturated fat more like that of saturated fat. Some disadvantages of them are that mono and polyunsaturated fats are healthier for the heart than saturated fats. In partial hydrogenation, trans fatty acids can form in the body and they're hard to metabolize. They accumulate in fatty tissue and are very difficult to excrete from the body. and they also increase levels of LDL cholesterol in our... What is that?
That's fine. We have to end the show, so finish what you want to say.
And it increases the LDL cholesterol in our low-quality energy source.
Sir, thank you for coming today. We hope to see you tonight in our evening news. Now we know our regularly scheduled programming is
It's already in progress.
Thank you. See, he doesn't even work here. The Mel TV, we don't talk mess, we stay.
Talk fast.
We don't talk mess. Sorry,
Yeah.
Frenchman.
We don't
Talk mess. We stay facts.
We state facts.
Amen.
Amen.
And now to our last category of the day, shelf life. Andrew, pick a question.
Question 1, please.
What is the meaning of shelf life? Go ahead, Steve.
All right. Shelf life is when food is no longer maintained the expected quality desired by the consumer because of changes in flavor, smell, texture, and appearance, or because of microbial spoilage.
Correct, Steve.
Of course. Effects on shelf life and quality of foods, please.
Explain the factors that affect the shelf life and quality of food. Andrew?
Is this working?
Go, yes,
Okay.
go on, Andrew.
Come on.
Andrew?
You know.
How much time do I have?
Just go on.
I know. Okay, well, some chemical factors that causes the decrease in the shelf life are the water content, chemical change like pH, light, and temperature.
Is that it?
I think so.
Wrong! You're supposed to explain. Water content is the loss of nutrients, browning, and rancidity. Dry foods become vulnerable microbial spoilage if they take on moisture. pH is off flavor, color change, browning, and loss of nutrients. Light is rancidity, vitamin loss, and fading of natural color. Temperature changes the rate of other forms of spoilage and contact with water. Next question, contestant 1.
Rancidity.
Describe the rancidity of fat. Go on, Steve.
All right. Rancidity is a perception of flavor in lipids, those that our senses perceive as off because they have a disagreeable smell, taste, texture, or appearance.
Good job. You actually have a brain.
I know.
Last question. Compare the processes of hydrolytic and oxidative rancidity in lipids.
All
Steve?
right. Hydrolytic rancidity is a breaking down of lipid into its components, fatty acids, and glycerol. It takes place more rapidly in the presence of enzymes, heat, and moisture. And I believe you deep fry something, water is present in food and high temperatures increase the rate of hydrolysis. Some examples are butanoic in rancid acids and butanoic acid in butter, I think. Also, oxidative rancidity is a fatty acid chains are oxidized typically by the addition of oxygen across the carbon-carbon double bond of the unsaturated fatty acid. And oily fish contain a high proportion of unsaturated fatty acids and are prone to oxidative rancidity. The process can be catalyzed by light and enzymes.
Awesome! Congratulations, contestant 2. You are the winner with a score of 25 and -5.
Yes! So what do I win?
You win recognition. Thank you. See you all next time.
How many bananas do we get?
Get 10, make sure they look good in color.
Do they have to look good?
Honey, food, as well as providing nutrients, it must be attractive. Color is an important property of food that adds to our enjoyment of eating when purchasing. See you later.
So how is the color able to appear on foods?
I overheard your question. I am Dr. Michelle's professor that specializes in the aspects of color. To answer your question, I must inform you that the color emitted is a direct result of the complementary color being absorbed. For example, as you can see, the red pigment of meat appears red because it absorbs the green light, its complement, and reflects the red light. Food can be naturally or artificially colored with a natural pigment or a
How can
dye.
you distinguish between a pigment and a dye?
A dye is a food-grade synthetic water-soluble colorant, and a pigment is a naturally occurring in plants and animals which can be either porphyrins, chronates, and anthocyanins.
What is all that stuff?
Well, anthocyanins are the most common in plants, responsible for the pink, red, purple and blue colors in fruits and vegetables like cranberries, blueberries, strawberries and raspberries. Chronoids are the most common in nature and are produced by algae. The chronoid acts as a precursor for vitamin A.
Honey, guess what I learned?
By seeing Edgar here, I can tell you that you learned a thing or two.
How do you know his name?
He was my first husband. I should have stayed with him. At least has the sense to pick good bananas.
What?
Well, did he mention that chlorophyll is the major light-harvesting pigments found in green plants, and that heme is the red pigment found in red blood cells and muscles tissue, and myoglobin is responsible for the purplish-red color of meat?
No, he did not, just like he did not mention he had a husband.
Well, honey, at least you learned something. Now you can expand your knowledge by being able to describe the factors that can affect the color stability of anthocyanins, carotenoids, chlorophyll, and even learn something new as heme.
Well, since I learned this much, why not, I guess? Probably I can learn about your past as well.
Don't be ridiculous. That's all in the past, geez.
Sure, well, continue.
Factors should include the effects of oxidation, temperature change, pH change, and the pressure of metal ions. You should analyze absorbance spectra that demonstrate these effects. Anthocyanins. In aqueous solution, equilibrium reaction excess between the 4 different structural points depending on the pH and temperature. They are most stable and most highly color at low pH and temperature. They found deeply colored coordination complex with iron +3 and aluminum ions as a source, which can be the metal cans to which the food is exposed, causes a discoloration in canned fruit. They become less stable when exposed to heat, causing a loss of color and browning. In carotenoids, the presence of multiple unsaturated carbon-carbon double bonds make carotenoids susceptible to oxidation catalyzed by light metals and hydrogen peroxide. Oxidation results in the bleaching of color, causing lots of vitamin A activity in DAW filters. They are stable up to 50 degrees Celsius in the pH range of 2 to 7 and, therefore, are not degraded by most forms of processing. With heating, the natural occurring trans isomer rearranges to the cis isomer.
Wow, you're still the amazing women I remembered. Smile, don't forget about how chlorophyll is affected. Reaction with heat depends on pH. In a basic solution, chlorophyll is stable. In an acidic solutions, chlorophyll is very unstable. When heated, the cell membrane of the plant deteriorates, releasing acids, which decrease the pH. This results in the magnesium atom being displaced by 2 hydrogen ions, resulting in the formation of olive brown pheophytin complex. This cell degradation during heating also makes the chlorophyll more susceptible to photodegradation.
How about ammonia? During oxidation, the oxygen binds to the purple red myoglobin, forming red hexamyoglobin. Oh. And during this process, iron is in the Fe+2 state, which can be converted to Fe+3, also known as myoglobin, when myoglobin and red hexamyoglobin are auto-oxidized, forming a brown-red color. This coloration can be minimized if the meat is stored in a place free of oxygen, removing the air out of the packages and injecting 100% CO2.
By the way, some synthetic dyes are known to be carcinogenic, so they have to be thoroughly tested before sending it out to the public.
Do you still want bananas? They might not look as brown.
Did you know there are 2 types of browning? There is non-enzymatic, or the Millard's reaction, and caramelization. In the Millard's reaction, a condensation reaction between an amino group on an amino acid or a protein and a reducing sugar like glucose or lactose affects the chemical composition of the foods. The amino acid lysine results in the most browning color, while cystine is the least. In caramelization, foods with high carbs, especially sucrose and reducing sugar, without nitrogen-containing compounds, are most commonly affected. The rate of reaction is increased by acid or base catalyzed at a pH of above 9 or below 3, a temperature above 120 C that occurs during baking and roasting of foods with high sugar content.
I almost forgot how intelligent you are, Edgar. Maybe we should go out for coffee afterwards.
I would love to.
Mark, go pay.
Genetically modified foods have increased in the past years. Today, a MLTV special will uncover what the fuss is all about. Genetically modified foods are known to have derived or produced from a genetically modified organisms. The food can be substantially substantially different from or essentially the same as conventional food in terms of composition, nutrient, taste, smell, texture, and functional characteristics. For instance, the first genetically modified bovine in 1990, a Belgian blue, was bred and able to produce cows that carry the lactoferrin production gene. He was modified to humanize his cow's milk. How is it significant? Today, Dr. Rapina from Empire University is here to discuss about the benefits and concerns of using genetic modified food.
Foods have benefited humanity in various ways. Such areas include crops, animals, and even the environment. Crops have been enhanced to taste better, a reduced maturation time, increase in nutrients and yield, improved resistance to diseases, pests and herbicides, enrichment of rice with vitamin A. In addition, animals have been genetically modified to increase its resistance, productivity, feed efficiency, better yields of milk and eggs, and improved animal health. In some terms of genetically modification to the environment, we can describe that as a friendly bioherbicide and bioinsecticide conservation of soil, water, and energy, improved natural waste management. Hey, make sure this food you pretend for today got good nutrients, had good color as well as good texture.
Joey, why is this thing texture important?
Oh, Janie, you's a woman, so you wouldn't understand this kind of things.
I can try.
Suit yourself. Well, you need to understand, texture is a physical property which foods carry. Many food ingredients are completely immiscible and so will form separate phases within the food, however. In some cases, the size of these phases can be very small, so as it appears homogeneous to the naked eye. A colloidal particle is many times larger than an individual molecule, but many times smaller than anything that can be seen with the naked eye. You gotta remember, a colloidal is a mixture of a dispersed phase and a continuous phase.
Like milk?
Quiet, Janie. You don't know. Well, you're right about milk. Milk is a great example of such dispersed system. It appears white because light scatters by protein and fat droplets.
So somewhere what you've seen. You saying is that the dispersed system is like kinetically stable mixture of one phase and another larger immiscible phase?
Woman, silence. You have no word, but yes, you is right. You don't know the types of dispersed systems that include suspension, emulsion, and the foams in food.
How can you tell them apart?
See, you don't know everything, but to answer your question, you are all colloidal mixtures and can be distinguished by the phase they present. Suspension, liquid continuous phase, soil dispersed phase. Example, I'd say probably molten chocolate.
I like chocolate.
Oh, thank you, Jane. Emulsion, there's liquid continuous phase and liquid dispersed phase. Examples, probably oil and water or milk, and water and oil or probably butter. And then there's also foam, which is a liquid solid continuous phase. Gas, dispersed phase as well. Some examples include whipped cream, eggs, egg whites, beer. You know I like that. Bread and I think meringue as well.
And you say because I is a woman, I would understand and have to learn.
You still haven't learned nothing, Janie. You still don't know the action of emulsifiers.
Well, hurry up!
Don't speak to me like that. Watch your mouth. Calm down, calm down. Let me tell you about emulsifiers. They help with the formation of emulsions and foams. They act as the interface between the liquid, solid, and gas phases in the dispersed system. And I think they're also soluble in fats and waters. To make an emulsifier, oil, water, and emulsifiers and mechanical energy, which is beating or mixing, are needed. Yes, I do. Are needed. Stabilizers prevent emulsions and foams from separating. Now, women are so unappreciated. Lord bless them.